All right, everyone stretch their legs. We can go on. There's a few worksheets that I want to proceed with. And these diagrams, I think everyone can understand now. We are repeating ourselves, but bear with me for this few minutes. And as I said earlier, you will really recognize these things in the exam. And I think you will answer the questions with much confidence. So we can proceed now, or proceed to the next worksheet. Just to emphasize on this one again, which we have done before break. Remember now, the process is transcription inside the nucleus and translation outside in the cytoplasma. The question on the other process that's taking place in the nucleus. This is protein synthesis. We know this is transcription, but the question was another process taking place in the nucleus. DNA molecules is duplicated. What is this? We call this DNA replication. What is the significance of DNA replication? Is we duplicate our genes to transfer to the next generation. But what is the significance of DNA in protein synthesis is the other question. It forms a template on which a messenger RNA is formed to form a specific protein. All right. So let's go on to a next worksheet. Now, in this case, you will see that they ask it different. Protein synthesis is asked so again, the diagram below shows a sequence of nitrogen base on a strand of DNA which codes for part of a protein molecule, but in all this is protein synthesis because we have codons or nitrogen base, we have amino acids. So let's look at how we're going to approach these type of questions. So this is not a diagram. They say that in this diagram below is a strand of DNA. Now this is important. Underline it and write down DNA so you don't forget this is a strand of a DNA. Now remember earlier on we said that we have a sequence. I'm going to show you. We have a sequence, and the sequence is we use DNA, messenger RNA, tRNA, and amino acid. That is our sequence. Now in this case, they give us the DNA. They give us the DNA, and they say the DNA in this sequence is G, T, adenine, thymine, guanine, and thymine, guanine, and guanine. They give us that. And now the question is, write down the messenger RNA. Now they want the messenger RNA. They give us the DNA, so now they want the messenger RNA. Now this is a tip again. If you get the answer like this, write down this order on a piece of paper or on your exam paper or wherever, and then it's very easy. Write down the messenger RNA codon on the sequence, so that is easy. Guanine binds to cytosine, thymine binds to adenine, thymine binds to adenine, right, from left to right. So I'm going to write 3.2.2 and I'm going to write it from left to right. Adenine, remember now, adenine, because it's mRNA, adenine binds on uracil. Thymine binds on adenine, guanine on cytosine. And then we have thymine binds on adenine and guanine, cytosine and cytosine. So 1, 2, 3 points. All right, from left to right, I write it, and that is my answer. Don't write this in your exam. This is the rough work. You only write that sentence. Only write the sentence, and that is what is on your answer sheet. So the next question then, they ask, the following diagram shows the anticodons of 9 different tRNAs, right? Anticodons. So they give us the anticodons, and it's tRNA. So this is a tRNA. This is my amino acid. This is tRNA. Amino acid, tRNA. And now they give us the question now. The question is, select and write down from above diagram the amino acids. They ask the amino acids, and they say that would be required for this basis sequence of mRNA. So this is mRNA. They give us mRNA. They give us mRNA. I'm going to write now here. We have DNA, messenger RNA, tRNA, and the amino acid. Okay. But what did they give us? They give us mRNA. They give us mRNA. And mRNA, which they give us is, let's do the first one, uracil, uracil, uracil. They give us uracil. Now they ask us, what is the amino acid? They want the amino acid at sequence. So what is the amino acid? Can I go and look where's uracil, uracil, uracil? No, I can't do it, and it's anyway not here. But I have to find the tRNA. I have to write down the tRNA. So now I must go and change uracil. Uracil binds with adenine, adenine. Adenine, is there adenine somewhere here? If I look, alright, there is it. So the first amino acid is phenylalanine or whatever. Alright, so I must write it in this sequence. So I'm going to write in the exam, on my exam pad or wherever, 3.2.3 in my exam book, and the first amino acid is phenylalanine. We write it out, and then the next one will be this one. So on your own, try to discover which amino acid will this messenger RNA code for. This one. Which amino acid will that code? You use again the sequence. Alright, let's do this. So they give us messenger RNA. They give us messenger RNA, and they give us guanine, uracil, and uracil. So we must write down what is the tRNA. Guanine binds to cytosine, uracil binds to adenine, uracil binds to adenine. So CAA. So now I look for CAA. There is CAA, and that is valine. Valine. Alright. And now my last amino acid that I want is this is the messenger RNA. The messenger RNA. We must write down the tRNA. So A binds to uracil, not thymine. Uracil binds to adenine, and guanine binds to cytosine. Because this is M, so mRNA. I write down the tRNA because tRNA is what they give me here. And uracil, adenine, and cytosine. Cytosine, and it is methionine. So there's my 3 amino acids that I write down, and 1, 2, 3 points. You will receive 3 points. So people, the most important thing in this approach, underline what they give you. They give you messenger RNA. They give you messenger RNA. You must work out what is the tRNA. because they also give you the amino acid. But you must write down what is the tRNA, because this is T. For example, if they said that this was also messenger, or this is mRNA, then you can work directly from here to there. But this is tRNA, and this is messenger RNA. So it's a difference. They give me messenger, they give me the amino acid. I must write down the tRNA, I write it down, and then I must see what amino acids bind there. In this case, the same. They give me DNA, they ask mRNA. So sometimes I can give you DNA, and they can ask tRNA. So there's my DNA, my messenger RNA, tRNA. So if they give me guanine, thymine, and thymine as my DNA, and they ask tRNA, don't write from there to there, go through the steps. Messenger RNA is guanine binds with cytosine, thymine binds with adenine and adenine, and cytosine binds with guanine, adenine binds with uracil, and uracil, remember uracil now because it's RNA, and that can't be T. So can you see, if you did directly from there to there, maybe you will make a mistake because you're going to write T binds to adenine. So first of all, go through the steps. But what did they give me? They give me DNA. In this case, they ask this one, or they can ask this one. But we go through the steps here. And this is a question that they love to ask. This type of question, it's a higher level cognitive question, and they ask that almost in every question paper, in section B, many times in section A, and then they give you an option A, B, C, or D, but you must go through the steps to get to your answer. So use your sequence in this question. Use your sequence: DNA, messenger, transcription, and amino acids. If you use your steps, there won't be a mistake in that. So let's do another one of these questions. So these questions. There's it again. All right, the question below are based on protein synthesis. Take this down. It's based on protein synthesis, and the question is: describe the role of DNA during transcription in protein synthesis. Describe the role of transcription and in protein synthesis. Transcription then is the first phase. It's in the cell the nucleus, and the role of DNA forms a template. DNA forms a template for RNA, and this template are arranged according to the DNA template to form a specific protein. Remember, to form a particular protein. Template for messenger RNA, the nucleus arranged from the messenger RNA. It's a complementary base, and it's for a particular protein synthesis. Right, question 4.3.2. Question 4.3.2. The diagram below shows a sequence of nitrogen base of a small part of a DNA strand, which codes for a part of a protein molecule. So this is a part of a protein molecule. They say it's DNA. So from DNA, you must write the messenger RNA. So what do? We say, right, DNA, messenger DNA, dan's tRNA en amino acid. What did they give us? They give us the DNA, so that is easy. They just want the messenger. We write from left to right, so in the exam pad you will write 4.3.2. From left to right, cytosine binds with guanine, guanine binds on cytosine, guanine binds on cytosine. Thymine binds at adenine, adenine binds at uracil, binds at adenine, and then we have cytosine that binds on guanine, guanine and thymine binds on adenine. Remember this one? This is where the most learners make a mistake, because they say adenine binds to thymine, but this is RNA. This is the RNA that they want. So that is why we have uracil. And we write it in this sequence, not from top to bottom, and you will receive your points, will be allocated like that. All right. Be careful what they give. They can give mRNA. They can say this is mRNA, and then they want DNA. So what did they give? They give DNA, and they ask mRNA. Sometimes they can give mRNA, and then they can ask DNA or tRNA. So make sure that you know what they give and what they ask. Question 4. Easy when I'm doing this. So I'm going to give you 5 minutes to do that question on your own or with your partner. Remember now, they show us tRNA, in this case, anticodons of tRNA. Remember, if they say anticodon, you must know that it is tRNA. They can only say that this was the anticodon, then you must know that it was tRNA. And they give you the amino acids, and they ask you to write down the sequence of the protein molecule. Remember now, this is the other catch. This is mRNA, and from mRNA you must convert to tRNA to get your specific amino acid. So a quick 2-minute time, 2 to 3 minutes, so you can do that quick. All right, the question, we must write the sequence of the amino acids. Remember, we are going to write from left to right, not from top to bottom. So they give us messenger RNA, guanine, 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 cytosine, cytosine, adenine, and adenine, guanine, uracil. So I take my sequence, my paper, and now I'm going to do the following. Messenger RNA, they give us guanine, they give us this sequence of messenger RNA. So from messenger RNA, I must convert it to tRNA, so I can connect it with amino acid. So guanine binds cytosine, guanine, cytosine, guanine, that is easy. So then cytosine binds on guanine, cytosine binds on guanine, and adenine, remember, binds on thymine. And now you can see it's now, I almost make a mistake, and that is what's happening with learners. This is easy, so we go adenine, and you can see I want to write thymine, but it's adenine is uracil, and guanine cytosine, and uracil is adenine. So please be careful with this. Be very careful with adenine and uracil in RNA. Adenine binds with uracil, guanine binds with cytosine, uracil binds then again with adenine. All right, and now I can go and I can seek my amino acids. CCC. CCC is glycine, and GGU. GGU is proline, and UCA is serine, that one in the bottom. and I write my proteins or my amino acids from left to right, not from the top to the bottom, from left to right, and that is the sequence. So people look at what they give you. Do they give you DNA and asked tRNA, or are they giving tRNA and ask mRNA? You write it down and then you work to that. If you have to work from DNA to tRNA, go through that step. If you have to work from tRNA to DNA, you work with that step also. For example, they can give you a enzyme. For example, they ask you valine. They say valine is a amino acid, and they want the DNA sequence of that, or the DNA basis. So we have mRNA, tRNA, and the amino acid. In this case, they give us the amino acid. The amino acid is valine, and we look at valine, we have CAA, so CAA is the T. From C we have to work now backwards. Can you see? So cytosine binds at guanine, adenine binds at uracil, adenine binds at uracil, guanine binds on cytosine, uracil binds on adenine, uracil binds on adenine. So that is my DNA sequence. Alright, so make sure what do they give you and what do they ask you, and use this order. Please use that order to answer your questions. That sequence is very, very important.